Students, today we will study the sexual incompatibility. In natural condition, wide variety of pollen grain alight on the receptive stigma. However, not all of them germinate nor bring about fertilization. If a carpel bearing the functional gamete is pollinated with the viable and fertile pollen yet fail to produce seed, the two individual male and female are said to be incompatible and the phenomena is termed as sexual incompatibility. Sexual incompatibility may exist between the member of different species, that is interspecific, or between the member of the same species, that is intraspecific. In intraspecific or self-incompatibility, a large number of angiospermic plants have the characteristics to be fertilized successfully only by the pollen of other plant. Perhaps the most effective and the natural method which prevents self-pollination is self-incompatibility. Based on the morphology of mating type, self-incompatibility may be of two types. Homomorphic, in a species when all the mating types are morphologically similar, it is said to be homomorphic type. Plants of this type are identified by breeding testing only. Heteromorphic type. In heteromorphic type refer to the condition where two or three distinct different morphological mating types are present within a species and may be identified without breeding test. The mating types usually differ in the structure and position of the sex organ, that is the stigma and the anther. Heteromorphic incompatibility may be regulated by one gene and the two allele as in primula, or by two genes with the two allele as in case of Lythrum. Genetic control of self incompatibility. Incompatibility is a gene control physiological process. Depending upon whether the genetic control is exercised by the male gametophyte or the sporophyte, self-incompatibility may be of the two types. They are the gametophytic self-incompatibility. The genotype of the male gametophyte determines the incompatibility process. Example in trifolium and the member of Gramineaceae, Liliaceae and Solanaceae. East and Maffelsdorf proposed the opposition S allele hypothesis, according to which incompatibility reactions are controlled by a single S gene having several alleles. If pollen grain possess the S allele common to any one of the two alleles present in the gametic cell of the female, the fertilization will not be effect between the two. This may be illustrated as when S1 and S2 sporophytic allele, they form the microsporogenesis pollen grain of S1 or S2 type. S1 and S2 they are incompatible. Ovary of S1 and S2 they are incompatible and there will be no fertilization. When the sporophyte is of S2 and S3, microsporogenesis is of S2 and S3, that means the pollen is of S2 and S3 and the ovary is of S1 and S2. 50% fertilization with the S3 pollen takes place. When the sporophyte is S3 and S4, microsporogenesis is of S1 and S4 type. S1 pollination S1 and S3 100% fertilization takes place when the ovary is of S1 and S2 type. Sporophytic self incompatibility. The process of incompatibility is controlled by the genotype of the sporophytic tissue. In such type a system, all the pollen behave in a similar manner irrespective of the S allele they carry and will be non-functional on S1 and S2 plant. This may be explained as when the sporophyte of S1 and S2, the microsporogenesis will be formed. The microsporogenesis will bear the pollen of S1, S2, S2, S3, S1, S3 or S1, S4, which pollinate the pistil of S1 and S2 and